Hello and welcome back everyone. My name is Shirsha and today we are going to be discussing about recombinant DNA technology. So recombinant DNA technology or RDT is one of the first things that you should be versed with when you start working in a lab. Because fine, we have talked about protein purification, we have talked about mammalian cell culture, we have talked about intricate experiments like FACS and flow cytometry, IHC and ICC, about Western blotting, co-IP and ELISA. But at the root of it, you require the DNA. Because as we know, DNA is the basic component that makes all of us up. And it is the thing that keeps the world running. Because even if you want to purify a protein and you say, but no, I don't work with DNA, I am a protein person. I purify protein and I am interested in structural things or the structural aspect of things. But then, in order to be able to produce that protein in the first place, you require its DNA. So, a knowledge of recombinant DNA technology or RDT is one of the basic things that you require to have, no matter what branch of biology you would be dealing with later, whether it be molecular biology or developmental biology or animal biology, whatever it is, a knowledge of the DNA and RDT is imperative for all of them. So, in this particular lecture, we are going to have a brief discussion about the origin of RDT and about restriction endonucleases, which is one of the crucial components or the crucial, well, I don't want to sound crude, but a crucial thing that is required for RDT. So, without further ado, let us dive into it. What is recombinant DNA technology? So it is the technology that allows manipulation of DNA in a living organism. So if you want any organism to express your protein of interest, then you should be able to manipulate that host organism's DNA, because this organism that you're using, most commonly a bacteria, will normally be not having the DNA for your protein of interest. It will not be expressing it under normal circumstances. So in order to get it to express the protein of your interest, you should be able to manipulate its DNA such that now it's either its genome contains your DNA of interest, and or you have this plasmid vector which contains your DNA of interest, which is present in the expression system and it is being expressed. So in RDT, so that is the abbreviation used for recombinant DNA technology, gene or DNA segments are isolated. So if you want to express and purify or study a specific protein, then you should first be able to identify and then isolate its gene. So you have the genetic map in your hand, because the gene is the blueprint of everything, right? So in RTT, DNA segments are isolated, they're amplified. Obviously, if you have a limited amount of the DNA, then that will not really suffice to serve all of your purposes. You can't perform all of your experiments with a limited amount of DNA. So you need to be able to amplify the DNA and then finally insert it into another cell with precision. Now, why with precision? Because let us say if so happens that when you're inserting your DNA, there's a mutation in a single amino acid residue. And this mutation is brought about by a single nucleotide mutation at the DNA level. So this one mutation can actually render your protein completely non-functional, completely useless. So precision is of utmost importance. Also, let us say that there is one deletion from one end, right? So as we know that three nucleotides code for one amino acid, and if there is one deletion, then it will just shift up the entire reading frame, and as a result the entire sequence of your protein would change. So precision is of, like I just said, utmost importance. RDT was made possible by the discovery, isolation, and application of restriction endonucleases back in the 1970s. So restriction endonucleases are the key players in RDT. Now RDT becomes useful only when the artificially produced recombinant DNA could be reproduced. That is, you can clone the DNA. So unless and until you can reproduce your recombinant DNA, it becomes useless because again you will run out of it. So DNA cloning is very, very important. And as I have discussed when talking about DNA isolation, that you one of the ways to amplify your DNA is to transform your plasmid vector containing your gene of interest into one of these bacterial strains like the H5 alpha, which typically maintain a high copy number of plasmids, and then the bacteria will simply amplify the plasmid thinking that it's its own, and you can then isolate the plasmid from there. So DNA cloning also is very, very important. Now, the recombinant DNA molecule is inserted into a host organism to produce new genetic combinations, and these new genetic combinations are of value to science, medicine, agriculture, and even industry. Recombinant DNA is the general name for a piece of DNA that has been created by combining at least two fragments from two different sources. So minimum you require two fragments of DNA, each of which have been isolated from two different sources, and when they are combined together, it is referred to as a recombinant DNA, and the technology behind it is called recombinant DNA technology. So, for instance, if you are inserting your gene of interest, which let us say you have isolated from a rat, and you are inserting it into a vector which is actually of bacterial origin. So, what you have now is a recombinant DNA. And just to be clear, what I mean by a vector, it is nothing but a plasmid which will act as a vehicle to carry your gene of interest and ensure its expression when transformed or transfected into an expression system. So recombinant DNA is possible. Why? Because DNA molecules from all organisms share the same chemical structure. That is, they are made up of only four nucleotides, A, T, G, and C. These four nucleotides make up the DNA in all living organisms, and that is the beauty of the system. That is what makes DNA recombination possible. And okay, so individuals or organisms only differ in the nucleotide sequence within that identical overall structure. And because the overall components or constituents are same, you can mix and match them as per your requirement. So recombinant DNA molecules are sometimes also called chimeric DNA because they can be made from material taken from two different species. So according to mythology, a chimera is an animal which has different parts of the body taken from different animals. So therefore, recombinant DNA molecules are also called chimeras because the constituent DNAs can come from different species. So now let us briefly talk about the history of restriction endonucleases. How did they come to be identified? So in the late 1960s, scientists Stuart Lynn and Werner Arber isolated examples of the two types of enzymes responsible for phage growth restriction in E. coli bacteria. So what is phage? A phage is a virus that attacks bacteria. So E. coli, as a part of its defense mechanism, developed these enzymes that would restrict the growth of these phages within them. What are these enzymes? So one of these enzymes methylated DNA, while the other one cleaved unmethylated DNA at a wide variety of locations along the length of the molecule. So the methylation serves as a protection. The DNA that is methylated would not be cleaved, and unmethylated DNA would be cleaved. So the first type of enzyme was called a methylase, while the other was called a restriction nuclease. Why restriction? Because it is restricting the DNA by cleaving it. These enzymatic tools were important to scientists who were gathering the tools required to cut and join DNA molecules. So technically or typically what the bacteria does is that it methylates its own DNA for protecting it, and then whenever this phage, which is actually invading the bacteria, injects its DNA into the bacteria, the host bacterium can now recognize the foreign DNA. Why? The foreign DNA is not methylated. So this methylation acts as a basis of distinction between self-DNA and foreign DNA for the bacteria. So it recognizes this unmethylated DNA and then cleaves it using this restriction nucleases. So because the restriction nuclease cleaves the DNA, and not only does it cleave the DNA, but it cleaves it at very specific sites. So this became important tools required for cutting and joining DNA molecules. So what was needed now was a tool that would cut DNA at specific sites rather than at random sites along the length of the molecule, so that scientists could cut DNA molecules in a predictable and reproducible way. So this specificity is required for ensuring reproducibility of the cleavage. Because if it's random cleavage, then there would be a lot of batch to batch variation and you can't really control it. But if it is sequence specific, then it becomes predictable. You can then design, oh fine, if I have so and so sequence, this enzyme can go and cleave over there. So a restriction enzyme or a restriction endonuclease is a specific endonuclease that is able to recognize specific short sequences of DNA and cleave the DNA either at or near the recognition site. So why do bacteria produce these restriction endonucleases? Like I already mentioned, it is a part of their defense mechanism. So restriction systems allow a bacteria to monitor the origin of the incoming DNA and to destroy it if it is recognized as foreign. Endonucleases that are primarily responsible for this degradation are called restriction endonucleases or restriction enzyme. Now one more point, why are these enzymes called endonucleases? Because they cleave within the DNA molecule. Enzymes that cleave at the edge of the DNA molecule would be referred to as exonucleases, hence the distinction. So restriction endonucleases, like I have already mentioned, are a part of the bacterial immune system. The host bacteria must protect itself from the degradative effect of the restriction endonucleases, and for that it needs to appropriately modify its own DNA, and it achieves that by methylating its own DNA. So modification that protects host DNA from degradative action of the endonucleases involves methylation of bases, primarily adenine or cytosine, at some specific sequences. And these specific sequences are expected to be absent or missing in the incoming foreign DNA. Otherwise, the incoming foreign DNA would also be methylated and it could not be cleaved and the entire point of the immune system would fail. So restriction endonucleases cleave double-stranded DNA, an important point to keep in mind. These enzymes are highly sequence specific, they exhibit great degree of specificity, and each enzyme acts at a limited number of sites in the DNA, which are called recognition or cutting sites. Now, if the DNA is digested by a restriction enzyme, the DNA will be then reduced to fragments of varying sizes, depending on how many cutting sites for that restriction enzyme are present in the DNA. So let us assume we have one circular closed double-stranded DNA. So if it is cut only once, that would linearize it. If it is cut twice, it would generate two fragments. If it's cut thrice, then more fragments, and so on and so forth. And the size of the fragments would depend on where within the DNA sequence the enzyme is specifically cutting. So let us now talk a little bit about how restriction endonucleases are named. I am pretty sure that all of you have heard the names of some common restriction endonucleases like EcoR1, BAMH1, HIN3. So how are they exactly named? There must be some logical reasoning behind it. It is not random. And indeed, there is some logical reasoning behind the naming of restriction endonucleases. So the names for restriction enzymes come from, first, the type of bacteria in which the enzyme is found. Second, the order in which the restriction enzyme was identified and then isolated. So because one particular bacterial species would definitely not have only one restriction enzyme. So the order in which it was identified and then isolated also plays a role in the naming of the restriction enzyme. So for example, EcoR1. This is the most common restriction endonuclease, and I'm pretty sure that almost all of you have heard its name. So how is it named? Let us break it up. So the R strain of Escherichia coli bacteria. So please pardon, E. coli should be in italics. There was an error in the editing. So E. co, E for Escherichia, co for coli. R denotes the strain of E. coli bacteria from which it was isolated, and 1 as it was the first E. coli restriction enzyme to be discovered. Okay, hence the name. So a fun activity would be for all of you to find out the reason behind the naming of some of these restriction enzymes that are commonly used. It's pretty straightforward. I will give you a hint. So for instance, BAMH1. So 1 indicates that it was the first enzyme to be isolated, or the first restriction enzyme to be isolated from this particular bacteria species. Look it up. It would be a fun exercise for all of you. Also, you would notice that most of the restriction enzymes have a 1 after their name, but there are some restriction enzymes which have higher number. Like for instance, you have EcoR1. So similarly, there also exists EcoR5. So now let us talk about the types of restriction systems. So 4 different types of restriction and modification systems have been characterized so far. These are type 1, type 2, type 3 and type 2s. So let us deal with them one by one. Type 1. So type 1 restriction systems comprise of one enzyme with different subunits encoded by the genes HSD-RMS for cleavage, modification and recognition. So all 3 functions, that is cleavage, modification and recognition, are all performed by one single enzyme. But this single enzyme has different subunits, and each of these subunits perform one of the functions. So one subunit performs cleavage, the other one performs modification, and the other one helps in recognition. Type 1 enzymes can recognize and methylate a single sequence. So it will recognize and methylate the same sequence, but cleavage occurs up to 1000 base pairs away. Methylation and cutting both require energy released from ATP. So because type 1 enzymes can cleave at any distance up to 1000 base pairs away from the recognition sequence, so exactly where it would cleave is not very highly conserved, and this would lead to some variation. So this would affect the reproducibility that we as scientists see. So therefore type 1 enzymes are not suitable for our purpose for using in recombinant DNA technology. Next, we come to type 2 restriction systems. So there are 2 different enzymes which recognize the same target sequence. So the target sequence that is recognized is the same, but now instead of one, two different enzymes are recognizing the same sequence. Now, the two enzymes can either cleave or modify the recognition sequence. So basically, one of the enzymes will cleave the sequence and the other one will modify the sequence. So it is possible to cleave DNA in absence of modification. Cleavage does not require any energy in the form of ATP. The enzymes recognize a defined, usually symmetrical sequence and cut within it. So type 2 enzymes, the sequence that they recognize is symmetrical in nature, and because they cleave within it, so this will give a reproducible pattern of DNA cleavage. Next, we have the type 3 restriction system. So this comprises of one enzyme with two different subunits. So just like type 1 system, the type 3 system also has only one enzyme with two different subunits. One for recognition and modification, and the other one is for cleavage. It will recognize and modify the same sequence, but cleaves 24 to 26 base pairs away. And because it does not really cleave within the recognition sequence, but rather cleaves outside of it, so this will again not really serve our purpose, because, well, you would then have to give extra DNA on either side of the restriction site where cleavage would occur. And then that has to be followed by your gene of interest. So this will simply make the entire process more messy. So it is avoided for the purpose of RDT. Finally, we have the type 2S system. So as the name suggests, it is like type 2 but with certain differences. So the recognition sequence for type 2S is asymmetrical. So for type 2, the recognition sequence is symmetrical. That is, if you just cut it from, well, if you come to the midpoint, then you read left to right on one side and it will just be the mirror image on the other side. I hope I'm clear. Well, let us say we have GAA, AAG. So that is a symmetrical sequence because GAA, now we have the midpoint, and on the other side you have the mirror image of that AAG. So that is a symmetrical recognition sequence. So for type 2S systems, the sequence that they recognize is asymmetrical. Cleavage occurs on one side of the recognition sequence up to 20 base pairs away. Again, because it is not within the sequence, so not really suited for us. So for recombinant DNA technology, the restriction enzymes that are used are type 2 restriction enzymes. Most type 2 enzymes recognize and cleave DNA within a particular sequence of 4 to 8 nucleotides, which have a twofold axis of rotational symmetry, that is, palindromic sequences. So what we talked about in the previous slide, the GAA-AAG, well, that sequence is not really palindromic because it does not have the rotational symmetry. So how would we define a palindromic sequence? A palindrome is a sequence of DNA that is the same when one strand is read from left to right or the other strand is read from right to left. That is, it comprises of adjacent inverted repeats. Okay, so this might seem more confusing. So let us clear it up a little bit more. A palindromic sequence is a nucleic acid sequence in a double-stranded DNA or RNA molecule whereby reading in a certain direction, say for instance from the 5' to the 3' end on one strand, is identical to the sequence in the same direction 5' to 3' on the complementary strand. So here when we say from right to left on the other strand, that essentially means 5' to 3' on the complementary strand. Why? Because, well, this is the left to right and this is the right to left, which is essentially the 5' to 3' direction of reading the sequence for both the strands. So this is a palindromic sequence. You see, you start from here, you have T, T, A, G, C, A, C. From here, you have the same T, T, A, G, C, A, C. So this is called a twofold axis of rotational symmetry, because if you rotate it about this point, so from this point you turn it like this and then you flip it, you will have the same sequence. Hence, a palindromic sequence. So let us talk a little bit now about the cleavage site and site of modification. So site of modification is a mark for the host DNA. It is for self-preservation. Site of cleavage is a mark for the foreign DNA. It is for protection and for fighting against the foreign DNA. For example, for EcoR1, this sequence GAATTC, you see it's a palindrome. On the other strand, it reads the same, GAATTC. So this is the palindromic sequence that is recognized by EcoR1, where the star indicates the cut site. So the enzyme will cut over here, whereas this hash or this hyphen indicates the modification site. I think there has been a mix-up. It's supposed to be the other way around. So the end product of EcoR1 cut DNA looks something like this. So because, yeah, there is definitely a mix-up. So the slash mark indicates the cut site and the star or the asterisk indicates the modification site. Please ignore this mistake. I'm sorry. So when you cleave it, you have something like this. So you see it has been cleaved over here and over here. So now these two DNA strands can separate from each other at this site of cleavage. So this cleavage, what it essentially does, it generates DNA fragments with 5' overhangs. Now why do we call it 5' overhangs? Because the excessive region or the overhanging region is on the 5' end. So these are called 5' overhangs. Okay, so it has cut over here at this G and this separates out. So the other strand, so the overhang would be on the, not on the strand which has been cleaved after the G, but on the other strand. See, here also it is cleaved after the G. This G is gone, so it has this overhang over here. And on the other strand, so this G was a part of this strand and this G is a part of the lower strand. So once that is cleaved, you have another overhang here. So both these DNA fragments have a 5' overhanging region. And such DNA fragments are said to have sticky ends. Now why are these ends sticky? So these ends, because now see over here, what is the sequence? It's AATTC and here you have this CTTAA. So essentially these are complementary to each other, and being complementary, they have a tendency of annealing back. So they will anneal, but there will be no ligation between the fragmented DNA. So because they have a tendency to anneal back, so therefore they are said to have sticky ends because they just stick to each other. So a little bit about blunt end versus sticky end. So DNA ends refer to the properties of the ends of linear DNA molecules, which in molecular biology are described as either sticky or blunt based on the shape of the complementary strands at the terminus. So what does that mean? In sticky ends, one strand is longer than the other, typically by at least a few nucleotides, such that the longer strand has bases that are left unpaired. And because they're left unpaired, whenever they encounter complementary sequences which are also unpaired, they will tend to anneal to them. And this is what makes them sticky. Whereas in blunt ends, both the strands are of equal length. That is, they end at the same base position, leaving no unpaired bases on either strand. And therefore, they do not have this tendency of annealing. Because if there is no unpaired DNA nucleotides, then what will anneal? you require unpaired nucleotides for annealing. So EcoR1 makes sticky ends. You see over here, it cleaves like this between after the first G from the 5' end. On both the strands, it cleaves like that. So you have the 5' overhang because this is the 5' end of the DNA. So these are called 5' overhangs. Whereas in blunt end, you see there are no overhangs. Both the top and the bottom strand are of the same length and there are no unpaired bases. So for ligation, that is for joining these two fragmented DNA strands together, sticky end ligation is better than blunt end ligation. Or if I reframe it, blunt end ligation is more difficult as compared to sticky end ligation. Because in blunt end ligation, the probability that these two ends will come in close enough proximity for the enzyme to act is lower. Whereas in case of sticky ends, because these are complementary to each other. See, this A is complementary to this T, this A to this T, set the second T. So, owing to this complementarity, sticky ends will anneal and this will bring the two DNA fragment ends in close proximation, which will allow ligation to occur more easily. Now, a couple of terminologies that we commonly encounter when dealing with restriction systems and restriction enzymes. So the first one is star activity. So it refers to the altered specificity of the restriction endonuclease due to lower ionic strength or altered pH. So generally restriction enzymes have very high specificity, but sometimes if there is an alteration of the pH or of the ionic strength, then this can lead to altered specificity. And such altered specificity is referred to as star activity. So for example, EcoR1 star cleaves at NAATTN after N, where N can be any base. So otherwise it is recognizing GAATTC and cleaving after the first guanine. But here the residue after which it is cleaving can be any base and the last base can also be any base. It doesn't really have to be necessarily cytosine. Next, we have isoschisomers. So isoschisomers are different restriction enzymes that recognize and cleave in the same location. So two different enzymes, they recognize the same location and cleave in the same location. They would be called isoschisomers. So for example, SPH1 and BBU1 are isoschisomers of each other. So you see, both of them recognize the same sequence CGTACG and both of them cleave before the last G. Next, we have neo-isoschisomers. So neo-isoschisomers are pairs of enzymes that recognize the same sequence but cleave it differently. So these are neo-isoschisomers. For example, SMA1 and XMA1. So both of them recognize the same sequence, triple G followed by triple C. But SMA1 cleaves in between the G and C, giving rise to blunt ends, whereas XMA1 cleaves after the first G, giving rise to 5' overhangs. Now, when will we have 5' overhangs? So, if the cleavage site is closer to the 5' end, we will have 5' overhangs because once it cleaves here, then this G will become the 5' end. Okay, and if the cleavage site was closer to the 3' end, then what would we have? What would be 3' overhangs? So a little bit more about restriction enzymes. So restriction enzymes could be either 4 base cutters, 6 base cutters, or 8 base cutters. So this naming is on the basis of what is the length of the recognition sequence. So for example, EcoR1 that we talked about, it recognizes the sequence GAATTC. So that is 6 nucleotides. So that is a 6 base cutter. So most of the enzymes that are commonly used are 6 base cutters. 4 base cutters and 8 base cutters are less commonly used. So some examples include, So for a 4-base cutter, you have these two enzymes. This is just an example. For 6-base cutters, you see KPN1, BGL2, the enzymes that you commonly find in a laboratory. These are 6-base cutters. Whereas 8-base cutters include not one. So what is the probability of the existence of a restriction site? So if we assume to begin with that the G plus C content is around 50% exactly. So for a 4-base cutter, the probability of a restriction site to exist is 4 to the power 4. That is, after every 256 base pairs, you can have a restriction site. But that is again just the mathematical probability. So why is it 4 raised to the power 4? That is because there are only 4 different nucleotides, A, T, G and C. So they have to exist in a specific combination, and because this specific combination is 4 residues or 4 nucleotides long, so it will be 4 multiplied by 4 into 4 into 4, right? So this is simply simple permutation combination. We wouldn't really say combination, this is simple permutation, right? So let us consider, for example, that you have this sequence G, A, T and C, the one recognized by South 3A1. So the probability that the first nucleotide would be G is 1 in 4. The probability that the second one will be A is again 1 in 4 because there are only 4 possible nucleotides. Same for T and C. So what you would have is 1 by 4 multiplied 4 times. So that gives you this probability of 4 to the power 4. Like the probability of this existing would be 1 in 256, or the other way around, it can be every 256 base pairs. I hope that's clear. In case you have any queries, do leave it in the comment section and I will try to clarify it. So similarly, for a 6-base cutter, it is 4 raised to the power 6, that is 4096. And for an 8-base cutter, it is 4 raised to the power 8, that is 65,536 base pairs. So longer the recognition sequence, lower is the probability of its existence and further apart it would be. Okay, now see, this point that I'm saying that further apart it would be is simply an probability assumption. It can be pretty close as well. So to better phrase it, I would say that shorter the recognition sequence, more frequent it would be. Longer the recognition sequence, lesser or lower are the chances of it existing naturally in any DNA sequence. That is the better way of framing it. So methylation affects cleavage. Yes, this is something we have already discussed, how methylation is a means for the system or the bacteria to protect its own host DNA. So two common methylating enzymes are DAM, which is the enzyme that transfers a methyl group from S-adenosyl methionine to adenine residues in the sequence GATC. So DAM is basically adenine methylase. And the other one is DCM, which is a cytosine methylase. So DCM modifies the internal cytosine residues in the sequence CCAGG and CCTGG. So the internal cytosine, that is this one, is the one that would modify. So this one. Okay. So in DNA with 50% GC content, the chance of having the above sequences is every 256 to 512 base pairs. Some or all sites for a restriction enzyme might be resistant to cleavage if the DNA is isolated from a strain expressing DCM or DAM methylases. Okay, so if you are isolating your DNA from such a E. coli strain which expresses DCM or DAM methylases, then you can't cleave it with the restriction enzyme because there is every possibility that the DNA would be methylated, and if methylated, the restriction enzyme cannot bind to it and cleave it. Therefore, most of the strains that are used in the laboratories for amplifying DNA, they are genetically modified to lack these methylases. They don't have them. They don't encode for them. So this is just a schematic representation of methylation of cytosine and methylation of adenine. So restriction systems should be eliminated in E. coli strains that are to be used as hosts. So E. coli strains that are used as hosts for recombinant DNA are usually made restriction deficient so that any foreign DNA, whether it be mammalian or plant, ligated into a plasmid vector, when transformed into the bacterial cell, will not be cleaved even if the foreign DNA has unmodified restriction sites. So this brings us back to our initial point that the entire point of knowing RDT and doing it is to get the E. coli system to express your gene of interest encoded in a plasmid vector. And your gene of interest can be isolated from any species. So unless and until the E. coli system is the restriction system deficient one, then it will recognize your DNA of interest as foreign and it will cleave it. So again, your entire purpose is defeated. So therefore, it is important for the E. coli strain to be used as an expression system to be restriction deficient. The host strain could still be modification proficient, and propagation on such hosts would allow the plasmid to get modified, and then subsequently the plasmid could be transformed to restriction proficient strains if required. So it is not mandatory for the strain to be modification deficient as well. It can be modification proficient, but it most definitely has to be restriction deficient. Under the circumstances where it is modification proficient, then when you propagate your plasmid in such a host, it will modify the plasmid as well, and then you can isolate this plasmid and express it in a restriction proficient strain. So this is the immigration control region of E. coli. So these are the genes. So we have the HSD RMS gene, right? So we have the MRR gene, which is involved in the acceptance of foreign DNA, which is modified. And we have the MRCC, MRCB. So you don't really need to know much about these genes in detail. Just know that these encode for both the modification as well as the restriction system. So, for example, the ECO-K12 restriction system encoded by the HSD RMS genes cleaves DNA that is not protected by methylation at the target site. So, this is actually the restriction system of the ECO-K12 strain, that is the E. coli K12 strain, and these genes cleave the DNA that is not protected by methylation at the target site. So therefore, strains like DH1 and DH5, which we commonly use in laboratories to amplify our DNA of interest and which are extensively used in cloning, have a mutation in this HSDR gene and therefore lack endonuclease activity. But it has these MCRC and MCRB genes, which are the modification genes and can therefore modify or still methylate the DNA. So just to give you an idea of what each of these genes are. So the MCRC is a modification gene or encodes for an enzyme that will modify these sites. So does the MCRB and so does the MRR. Whereas the HSD-M region that also encodes for a methylase subunit, and the HSD-S and HSD-R genes, they actually encode for the endonucleases. So that is it. That was our brief discussion and introduction on recombinant DNA technology, what it is, why we need to perform it. We talked about one of the basic and most important tools required for RDT, and that is restriction endonucleases. We briefly discussed about the different restriction endonuclease types, and we also briefly discussed about how the restriction endonucleases were discovered and that they are a part of the host or the bacteria's own immune system. We also talked about how these restriction systems have to be modified in order to allow these E. coli strains to be used in laboratories for our purposes. So that is it. I hope it was clear. In case you have any questions or any doubts, please feel free to leave it in the comment section and I will definitely get back to you with the answer. If you like this video, then do like it and share it. And if you're looking for more such content on different techniques used in laboratories and the principles underlying them, then do subscribe to the channel. And I will see you again next time. Bye.